When we think about medicines, we often imagine simple pills that can be easily manufactured and copied. But modern healthcare has moved far beyond that. Today, many of the most advanced treatments are biologics, complex therapies that are very different from traditional drugs. To understand biologics, we first need to look at how they differ from small molecule drugs and why biosimilars are not the same as generics. Let's begin with small molecule drugs. These are the medicines most people are familiar with, like paracetamol or ibuprofen. They are chemically synthesized, relatively small in size, and have well defined structures. Because of this simplicity, they can be exactly replicated. When their patents expire, other manufacturers can produce identical versions known as generics. Generics are approved based on bioequivalence. This means they must demonstrate that they release the same amount of active ingredient into the bloodstream at the same rate as the original drug. Since the molecule is identical, there is no need for full clinical trials again. But biologics are fundamentally different. Biologics are large, complex molecules produced using living systems such as bacteria, yeast, or mammalian cells. These include monoclonal antibodies, vaccines, and recombinant proteins like insulin. Unlike small molecules, biologics are not made through simple chemical reactions. They are grown and processed in controlled biological environments. Because of this, variability becomes a key factor. Even minor changes in manufacturing conditions like temperature, cell lines, or purification steps can influence the final product. This makes it impossible to create an exact copy of a biologic. And that is where biosimilars come in. A biosimilar is a product that is highly similar to an already approved biologic known as the reference product. The goal is to ensure there are no clinically meaningful differences in safety, purity, or potency. However, proving this similarity is much more complex than demonstrating bioequivalence. Biosimilar development follows a stepwise approach. It begins with extensive analytical studies comparing structure and function. This includes advanced techniques to assess protein structure, folding, and biological activity. Next comes non-clinical evaluation followed by clinical studies. These trials are not designed to reprove efficacy from the beginning, but rather to confirm that the biosimilar behaves the same as the reference product in terms of safety and effectiveness. Now let's look at the regulatory pathway, starting with the United States. In the US, biologics are regulated by the US Food and Drug Administration and approved through the Biologics License Application or BLA. For original biologics, companies submit a full application under Section 351A, including complete data on safety, purity, potency, and manufacturing. For biosimilars, there is an abbreviated pathway under Section 351K introduced by the Biologics Price Competition and Innovation Act. This allows developers to rely partly on existing knowledge of the reference product, reducing duplication of studies. However, the standards remain strict. The FDA uses a totality of evidence approach, evaluating analytical, non-clinical, and clinical data together to ensure high similarity. One important concept in the US is interchangeability. A biosimilar can be designated as interchangeable if it meets additional requirements, including evidence that switching between the biosimilar and the reference product does not affect safety or efficacy. This allows pharmacists to substitute it without consulting the prescriber. However, achieving this status requires additional studies and is more demanding. Now let's move to Europe. The European Union has been a pioneer in biosimilar regulation, with approvals handled by the European Medicines Agency through the centralized procedure. Europe approved its first biosimilar in 2006 and has since gained extensive experience. Like the US, the EU requires a comprehensive comparability exercise. However, it often places greater emphasis on analytical data. If similarity is strongly demonstrated at this level, the need for large clinical trials may be reduced. A key difference lies in substitution policies. Unlike the US, the EU does not grant a centralized interchangeability designation. Instead, decisions about substitution are made at the national level by individual countries. Despite this difference, biosimilar adoption in Europe has been strong due to regulatory confidence and long-term clinical experience. So what does all of this mean? Biologics are among the most advanced therapies available today, but they are also expensive. Biosimilars help introduce competition, reduce costs, and improve patient access to life-saving treatments. At the same time, their development is scientifically complex and requires rigorous regulatory oversight.
Subscribe to MedPharma Global and follow us on LinkedIn. Join our network for the latest in pharmaceutical innovation. Subscribe and follow us today.